Welcome to Spring LIB.
Hello everyone, I hope all is well, and today I'll be helping you with the 74th problem of the Chapter 14 problem set. And so A is asking you to explain how point mutations in mitochondria DNA can result in a loss of function in the critical cellular components such as cytochrome C, yet not be lethal to the cell. So cytochrome C is a protein within the mitochondria. So basically what I'd say is when DNA is damaged, so like say you have DNA and then you have obviously it's not going to look like that. It's going to be circular, but there's something wrong with it, so it cannot function. The proteins are not produced correctly, so maybe the protein is supposed to look like that, but then the protein is now turning up like that, and it's not working correctly. It is not lethal to the cell because there are so many more mitochondria, and also there are things within the cell like particular organelles to get rid of those dysfunctioning parts. And then you have B, which says oocyte mitochondria are randomly segregated during meiosis, resulting in variation of frequency of mitochondria DNA of the offspring. So explain how a loss of function does not accumulate, lowering the metabolic performance from generation to generation. So what happens is that more mitochondria are created when the cells are going through mitosis, when there's a sperm meets an egg, and both of these have variation of mitochondria. And what can happen is that as they reproduce or go through mitosis, it'll help with more mitochondria coming to be. So then you have not E, but actually you have C, which is analyze the data to draw alternative conclusions regarding the three fossils. So you have the Sima, and then you have the Dinosaurians, and then you have Neanderthals. So this one is due to alleles, and then you have this one due to you could do common amounts of ATCG. So as you know in the Sanger method, that if there's similar amounts of nucleotides, and they're particularly always closely related. So that's how I'd do it. And then the alleles is because there were similar alleles passed on, so meaning that they had a common ancestor. So now you have D, which says design a plan to differentiate or resolve these alternative conclusions. So basically for the Neanderthals, what I'd do is I'd sequence it and see how much ATCG, so the nucleotides I get, to conclude that they are from a similar species, because if they're from a similar species, then this would equal the Sima fossil. Well, not completely equal, but it'd be very similar in range. And then for the Dinosaurians, basically is the alleles. So if they're similar and they have similar traits, then obviously they'd come from a similar ancestor. So I hope you found this helpful, and I hope you have a great day. Thank you.